But this process continues during fourth week as well. And during fourth week, somites are formed and then they undergo further differentiation. Each somite divides into parts, sclerotum and dermamyotum, and sclerotum gives rise to vertebra, and dermamyotum gives rise to the muscle and dermis of the skin. And as body of vertebra form around the notochord degenerates and exists only in the intervertebral discs. Formation of neural plate also occurs during third week. This structure appears as a thickening of the ectoderm at day approximately 17. But fusion of neural folds begins at day 22. This is first day of fourth week. And closure of neural tube occurs during fourth week. So you can see here that uh, neural tube is closing. Closure begins in the future occipital region, and this process occurs in both directions, craniocaudal directions. But neural tube is still open at cranial and caudal ends, and these openings are called neuropores. You can see cranial neuropore or rostral neuropore and caudal neuropore. And uh, neural tube communicates with amniotic cavity through these neuropores. Cranial neuropore begins closure at day 24. So at day 24, cranial neuropore is closing, but caudal neuropore is still open. At day 26, caudal neuropore is closing and cranial neuropore is already closed. So by the end of the fourth week, both neuropores are closed normally. If there are some disturbances during closure of neural tube, they may cause very severe birth defects such as microencephaly or spina bifida. So this is very critical period of human development when closure of neural tube occurs. And as you already know, neural tube gives rise to central nervous system, brain, and spinal cord. What is a critical developmental event during fourth week? This is morphogenesis. You already know what is morphogenesis. Can you tell me what is morphogenesis?
Teacher, it's the formation of the three germ layers.
No, this is
Differentiation.
calculation, Natalie. I asked you what is morphogenesis. Is
Beginning of formation the body of the embryo.
formation of the shape of the embryo.
Yes.
Establishment of body form is morphogenesis, and in morphogenesis, a very important event is folding. Folding occurs in median plane and in horizontal plane at the same time. And during folding in horizontal plane, lateral folds are produced. But after folding in median plane, head and tail folds are produced. And after folding, flat embryonic disc is converted into cylindrical embryo. As you remember, during second and third weeks, embryo is almost right. It's flat. It's flat relaminar disc, but after folding, it becomes C-shaped, cylindrical structure. And now we will describe folding of the embryo in both directions. So you can observe here neural tube. You know that neural tube enlarges during fourth week. This region is cranial region. It's broad. Folds in this region are thicker than in caudal region. And this is a developing brain. And at first it projects into amniotic cavity, dorsal in the amniotic cavity, but then it grows, it overhangs oropharyngeal membrane. So, development of brain and elongation of the embryo results in cranial folding. Pay attention how position of structures changes after folding. What is position of the structures before folding? This is oropharyngeal membrane. Heart tube lies cranial to it, and septum transversum, this region of mesoderm, which is called septum transversum, lies cranial to heart, but these structures move ventrally. And after cranial folding, heart lies caudal to oropharyngeal membrane, and septum transversum lies caudal to the heart. The septum transversum develops into the diaphragm. So you should compare what was position of structures before folding and after folding. They move ventrally. So head fold is formed, and part of umbilical vesicle and endoderm germ layer gets incorporated into the embryo as a foregut. So this is a foregut. You can observe here formation of foregut. This yellow layer is endodermal germ layer, and it gets incorporated into the embryo as a foregut. Foregut is primordium of esophagus and pharynx. Let's describe folding of the caudal region of the embryo. This is um, neural tube, but it's not cranial part. It's a distal part of the neural tube, which is narrow part, and it's primordium of spinal cord. It grows, elongates, and results in folding and formation of tail fold. You can observe here formation of tail fold. Now position of structures. This is remnant of primitive strip, and it lies cranial to cloacal membrane before folding. But after folding, as you can see, primitive strip lies caudal to cloacal membrane. So these structures change in position. Uh, what about connecting stalk and allantois? Connecting stalk moves ventrally, and now it is attached on ventral surface of the embryo, and allantois gets incorporated, partially incorporated into the embryo. Connecting stalk is primordium of umbilical cord. Amnion envelopes connecting stalk and forms epithelial covering of umbilical cord. So this is future umbilical cord. Part of the umbilical vesicle and endoderm is incorporated into the embryo as a hindgut. You can see here hindgut, and part of hindgut dilates to form cloaca. Cloaca is primordium of urinary tract. This is a diagram. Now we can observe this process with electron microscope. So this is hindgut. So, as you can see, after folding in medium plane, uh, after cranial and caudal folding, head fold and tail fold are formed. And now embryo is not dried, it's carved, it's C-shaped. But at the same time, simultaneously, folding occurs in the median plane. And after this, lateral folds, left and right lateral folds are formed. Part of the umbilical vesicle is incorporated into the embryo as a midgut. So lateral folding completes the gut tube. This is midgut, and it is primordium of small intestine. Pay attention that before lateral folding, intraembryonic cellum communicates widely with extraembryonic cellum, but after lateral folding, this communication reduces. You can see here intraembryonic cellum, and it does not communicate with extraembryonic cellum after lateral folding. Pay attention that connection between umbilical vesicle and midgut was wide before lateral folding, but during folding it reduces to a narrow channel which is called omphaloenteric duct. So after lateral folding, embryo is cylindrical. You can see surface ectoderm, neural tube under surface ectoderm, intraembryonic cellum, and gut. So let's describe all structures. You can see section through the cylindrical embryo, blue layer, of course, it's surface ectoderm. Neural tube lies under surface ectoderm, and you can observe not occurred under neural tube. This is aorta and this is gut. Now what can you see on each side of neural tube and not occurred? Somites, so segments of paraxial mesoderm. Then comes next region of mesoderm, which is called intermediate mesoderm, and then you can see two layers of lateral mesoderm. This layer is splanchnic mesoderm. It surrounds primary gut. And another layer is somatic layer. It is adjacent to ectoderm and forms the body wall of the embryo. So, as you can see, establishment of body shape, body form occurs during fourth week, and it results from folding of the embryo in cranial, caudal, and lateral direction. So first of all, you should study folding of the embryo. Now you can read these titles together with me. Uh, you can see that neurulation is not completed yet because neuropores are still open. Uh, it's the same week of development. You can see that neuropores are still open, but amniotic membrane and the sac was removed, and you can see gut. You can see midgut. And now you can see ventral view of midgut. So, which structures develop during fourth week? Pharyngeal arches. So, this is fourth week. First and second pharyngeal arches appear at day 24, but at day 26, three pairs of pharyngeal arches are visible. You can see pharyngeal arches better here. First pharyngeal arch is called mandibular arch because the major part of this arch gives rise to mandible, lower jaw. But part of this arch gives rise to maxilla, upper jaw. So first pharyngeal arch gives rise to the maxilla and mandible. Second pharyngeal arch elongates, it overgrows third and fourth arches and gives rise to a cervical sinus during fifth week. You can see here groove between first and second pharyngeal arches, and this groove develops as an external acoustic meatus, and swelling small hillocks develop around this groove. They are called auricular hillocks, and they contribute to formation of auricle of the ear. So auricle and external acoustic meatus develop during 6 week. What is this? This is heart prominence. It's large, uh, and this long tail-like region is called caudal eminence. So during fourth week, you can see also upper and lower limb buds. During fifth week, changes are minor compared with fourth week, but head enlarges and it contacts now with heart prominence on the ventral surface of the embryo. You can observe lens placode on each side of the head. What are lens placodes? They are ectodermal thickenings, and they are sites of development lenses of ear, eyes, lenses of eyes develop in this region. So you can see here upper limb bud and lower limb bud. One is pedal shaped, another is flipper like. This is umbilical cord. What is the most important developmental event during 6 week? First of all, differentiation of upper limbs. So upper limbs show differentiation during 6 week, and different regions, for example, wrist, elbow, shoulder are visible, and hand plates develop during 6 week. And digital rays develop in the hand plates. What are digital rays? They are primordia of fingers. Pay attention that development differentiation of lower limbs begins later, after 5 days approximately, and regions of lower limbs differentiate during 7 and 8 weeks. When ossification of bones begins, during seventh week, ossification of upper limbs begins, but in lower limbs, ossification of bones begins in the eighth week. So eighth week is the last week of embryonic period of human development. During eighth week, ossification begins in the femora. All regions of upper limbs and lower limbs are already apparent. Fingers and toes are separated. Pay attention that this long tail-like caudal eminence disappears embryo straightens and it has distinct human appearance now, but head is still larger compared with trunk. Pay attention low position of external ear. Notes the low position of external ear. Eyelids develop, and what is also characteristic feature of this period that vascular plexus develops around the head. Scalp vascular plexus around the head is characteristic feature of ACE week. So this is embryo during this is beginning of ACE week, and by the end of the ACE week, day 56, it has distinct human appearance. So most organ systems begin to develop during this period, 4 to 8 weeks, and that's why this is very critical period of human development, and exposure of the embryo to the teratogens such as drugs or viruses may cause different birth effects. So now we have few minutes and I will show you another slide demonstrating differentiating of somites. I told you that they give rise to sclerotum and dermamyotum, but I want to show you another presentation. It will help you to understand this process. Wait a minute, please. Just a moment. Development definition and going to go to show. Why I can't find this presentation? Okay, now I will do it once again. Okay, can you see it now?
Yes,
Can
doctor.
And do you remember what I told you a few minutes later? These are some segments of paraxial mesoderm. They are located on each side of notochord. This is notochord. And as I told you, each somite gives rise to sclerotum and dermamyotum. This is sclerotum, this is dermamyotum. Each sclerotum divides into halves, cranial and caudal. Now look here how each vertebra is formed. Caudal part of one sclerotum fuses with cranial part to adjacent sclerotum to form vertebra. So, two adjacent sclerotums contribute to formation of each vertebra and so on down the spine. This is neural tube and you can see nerves, they exist between vertebrae. Dermamyotum, this is dermamyotum. It divides into parts, dermatome and myotome. Dermatome gives rise to dermis of the skin, and myotome gives rise to muscles. That's why we told that somites give rise to exoskeleton, associated musculature, and dermis of the skin. And as bodies of vertebrae develop around the notochord, it degenerates and it exists only in the intervertebral discs. So you can see here vertebra now, and intervertebral discs as well. And this region, which is called nucleus pulposus, is a remnant of notochord. So this is a notochord. You can see here sclerotum, which forms vertebra around the notochord, and remnant of notochord exists as nucleus pulposus in the intervertebral discs. There are four sclerotums in the occipital region. They fuse to form occipital bone. There are eight sclerotums in the cervical region, and they give rise to seven vertebrae. Why? Because cranial part of first cervical sclerotum fuses with occipital bone. And first cervical vertebra, which is called atlas, is formed by fusion of caudal part of first and cranial part of second cervical sclerotus. So you can see here vertebra and spinal nerve. So that's all for today. I think it's enough for today.
Okay, doctor.
So what is your homework for next lesson? Chapter 5.
The full chapter 5, the whole.
Chapter 5. Still,
Doctor, can you please send us this PowerPoint? Like,
of course I will send it.
because we always
Wait,
have this PowerPoint before like the class in one day.
what? I
We always
always send presentations to all groups. I will send it now after class.
Okay, doctor, thank
Okay,
you.
doctor, thanks.
So check it, please.
Thanks.